Today we are going to learn all the steps of Krebs cycle in a very funny and in a very amazing manner. Apart from calling it as the Krebs cycle, we can also suggest, suppose that is citric acid cycle and tricarboxylic acid cycle. This step is known as SLP step, which is substrate level phosphorylation. Why? Hello and welcome to all my dear students. Today here I am to discuss one of the hottest topic of class 11th biology, that is Krebs cycle. Today we are going to learn all the steps of Krebs cycle in a very funny and in a very amazing manner. So before we start with the actual steps of Krebs cycle, first let me tell you that where this process takes place. So the process of Krebs cycle occurs inside the matrix of mitochondria. This whole concept of oxidation of acetyl coenzyme A into CO2 was explained by Sir Hanslet Krebs. That is why the name is Krebs cycle. Apart from calling it as the Krebs cycle, we can also suggest, suppose, two more names to it, that is citric acid cycle and tricarboxylic acid cycle. Now, while teaching you the steps of Krebs cycle, we will learn one by one that why these other names are also given to Krebs cycle. So, are we ready? This is the screenshot of your NCERT explaining the Krebs cycle in the simplest possible manner. So we are also going to do in the same way. So first of all, beta, whenever you study Krebs cycle, always try to mention link reaction also. Why? Because the first stable, or you can say that the first substrate of Krebs cycle is acetyl coenzyme A, a 2-carbon compound that actually comes from link reaction. So directly, instead of showing condensation of acetyl coenzyme A with oxaloacetic acid, it is always suggested to start from pyruvic acid. That means from link reaction, first we are going to produce the first substrate of Krebs cycle, which is acetyl coenzyme A, and then we will start the actual process of Krebs cycle. So first of all, here you can very clearly see that pyruvic acid in the matrix of mitochondria undergoes oxidative decarboxylation, which means this pyruvic acid will remove its CO2, will undergo oxidation via losing its electron and proton to NAD positive. NAD positive accepts the electron and proton and get reduced to NADH2 while oxidizing pyruvic acid into acetyl coenzyme A. To this pyruvic acid, we also attach coenzyme A. So after doing 3 steps, this pyruvic acid is finally converted into acetyl coenzyme A. This whole step of link reaction, remember guys, remember students, it is catalyzed by the enzyme pyruvate dehydrogenase, right? Okay, now you have obtained acetyl coenzyme A. Now it's the time to learn that how this acetyl coenzyme A gets completely oxidized to release CO2 molecule. So now I am going to start from acetyl coenzyme A because link reaction we have already discussed before. So what do you have? You have a 2-carbon compound which is acetyl coenzyme A that came from link reaction, same in the matrix of mitochondria. Now this acetyl coenzyme A, a 2-carbon compound, attaches to or condenses with a 4-carbon compound which is oxaloacetic acid. This is 4 carbon, so 4 plus 2, it yields the first stable product which is a 6-carbon compound, that is citric acid. Since the first stable product is a 6-carbon compound, that is why this Krebs cycle is also referred to as citric acid cycle. Okay, so we call it as Krebs cycle because of the scientist name. We can call it also as a citric acid cycle since the first stable product is citric acid. Now, the 6-carbon compound citric acid undergoes isomerization into another 6-carbon compound which is isocitric acid. So, normally isomerization takes place. That means adjustment of the carbon atoms, hydrogen and everything takes place. And this citric acid is converted into isocitric acid. Okay, so this is isomerization that takes place in the presence of the enzyme isomerase. Over here, the enzyme is citrate synthetase, because we are synthesizing citric acid. Now, this 6-carbon compound isocitric acid undergoes oxidative decarboxylation, like you have already studied in case of pyruvic acid. Okay, so first of all, it is going to remove its CO2. That means decarboxylation is being done. And then oxidation. Oxidation how? So NAD positive again over here is going to act as an oxidizing agent, which will extract electron and proton from isocitric acid, and thus it will get oxidized. However, NAD positive, after accepting electron and proton, this gets reduced to NADH2. Now, after oxidative decarboxylation, basically beta, this isocitric acid converts itself into alpha ketoglutaric acid. Now, how many carbon left? 6, out of which 1 is being removed in the form of CO2. So, how many left? 5. So, therefore, this isocitric acid is now a 5-carbon compound, which again will undergo oxidative decarboxylation. So, now, again, this is going to undergo oxidation via same method, that is removing proton and electron. And the oxidizing agent is again going to be NAD positive. So, this is going to get oxidized into NADH2. Correct? Parallelly, it will also remove 1 carbon in the form of CO2. So, now what product are you going to obtain? Plus, addition of coenzyme A also takes place. So, now which intermediate product are we going to achieve over here? So, this alpha ketoglutaric acid, which is a 5-carbon compound, now has released 1 carbon in the form of CO2. So, now 4 carbon left, and hence a 4-carbon compound, succinyl coenzyme A, will be formed. Right? So, how many intermediates you have seen so far? You started with citric acid, isocitric acid, alpha ketoglutaric acid, succinyl coenzyme A. This succinyl coenzyme A has lot of energy trapped inside it. So, what this succinyl coenzyme A will do, it will not only release its coenzyme A, but it will also remove one of its terminal phosphate and will give it to GDP, guanosine diphosphate. So, guanosine diphosphate over here will accept the phosphate group from the succinyl coenzyme A and will convert itself into energy intermediate, which is GTP. This step is known as SLP step, which is substrate level phosphorylation. Why? Because over here you are observing the formation of energy at substrate level. So, succinyl coenzyme A, after producing GTP, energy in the form of GTP, it will then convert itself into another 4-carbon compound, succinic acid. Now, this succinic acid will now get further oxidized, but this time oxidation will occur with the help of another redox equivalent, that is FAD positive. So, FAD positive will accept the free electron and proton from succinic acid, will get reduced into FADH2 while oxidizing succinic acid into another 4-carbon compound, that is fumaric acid. Correct? This fumaric acid will continue and it will get converted into malic acid in the presence of the enzyme fumarase. So, fumarase enzyme will be involved over here to convert fumaric acid into malic acid. Finally, malic acid, in order to maintain the continuity of the cycle, will finally convert itself into the primary acceptor of acetyl coenzyme A, which is oxaloacetic acid. And in order to convert itself into oxaloacetic acid, malic acid undergoes simple oxidation with the help of NAD positive. So, here also beta, NAD positive will get reduced to produce NADH while converting malic acid into oxaloacetic acid, thus completing the Krebs cycle. So here you have observed that per Krebs cycle, in order to oxidize a 2-carbon compound acetyl coenzyme A, we have produced 1, 2, 3 molecules of NADH, 1 molecule of FADH2 over here, plus 1 direct source of energy, which is GTP. Apart from this, we have also generated 2 molecules of CO2. Yes or no? So this is whole concept of Krebs cycle, where in order to oxidize 1 molecule of acetyl coenzyme A into 2 molecules of CO2, this whole drama takes place inside the matrix of mitochondria. Now, the catch is to learn the substrate name. The catch is to learn all the intermediates of Krebs cycle in a sequence. While you people attempt the test, so many intermediate steps are asked in your examination, yes or no, and usually students do get confused that was it isocitric acid first or was it citric acid first. So now here I am going to share you an amazing mnemonic. If you remember that, then all the steps, all the intermediates will be absolutely forever clear in your mind. So my simple mnemonic says, can I keep selling sweets for money, officer? So I'm asking an officer that can I keep selling sweets for money, officer? Now see, is it very simple now? I'm just asking a question to an officer that can I keep selling sweets for money, officer? Simple. Can you people learn this mnemonic? Now see how this mnemonic helps you to learn the intermediates of Krebs cycle. See over here, beta. Can ka C, that stands for citric acid. So again I am going to write it over here for everyone. Pay attention. So my mnemonic says, can I keep selling sweets for money, officer? Many of the students must have already decoded this mnemonic. Yes or no? Very, very easy, isn't it? So first C stands for citric acid, which is the first stable product. Okay, followed by I, isocitric acid. Citric acid isomerizes into isocitric acid. K stands for alpha ketoglutaric acid, right? So isocitric acid undergoes oxidative decarboxylation to produce alpha ketoglutaric acid. S stands for succinyl coenzyme A. Again, oxidative decarboxylation takes place. Next S is for succinic acid. Here substrate level phosphorylation takes place. F stands for fumarase, simple oxidation. M stands for malic acid, and last but not the least, O stands for oxaloacetic acid. Isn't it very, very simple? So here what is taking place? Isomerization. Here and here, in both the steps, oxidative decarboxylation is taking place. Yes or no? Even like that you can write it and learn it forever. Here, substrate level phosphorylation, SLP step, where energy is generated. So substrate level phosphorylation. Next, fumaric acid. Simple oxidation is taking place with the help of FAD positive. Then fumaric acid to malic acid, dehydration, removal of water is taking place, and last but not the least is again oxidation step via NAD positive. So isn't it not easy to learn the Krebs cycle? Yes or no? So please do mention in the comment section that did you people enjoy this whole mnemonic? If yes, then this whole video is very successful. Now, apart from giving you this amazing mnemonic to learn your Krebs cycle, I have a very exciting offer for you all. If you people are the students of class 11th, maybe JEE or NEET, and still looking for that how to prepare for your NEET and JEE respectively, then don't worry beta, we have a special offer for you all to join Arjuna NEET/JEE 2027 batch. Now, there are two types of offers for the same batch. One is the simple, that is you get the access to the batches, you get the access to the PDF and notes, etc. Now, if you are going to take the batch which is the only simple batch, then that is going to cost you, that initially it used to cost 6,300, but now as an offer the price has been reduced to 3,700. However, if you want some additional feature apart from the regular classes, regular DPPs and PDF notes, if you want some extra additional features, then you can take the Infinity Pro batch of Arjuna 2027 for both NEET and JEE. Initially, it used to cost 8,700, but now for you all, the price has been reduced to 4,800 only. So what are you all waiting for? Go check the batch, get yourself enrolled, and still you have the opportunity to begin your preparation. Thank you.